Welcome everybody, and welcome to today's masterclass, Info Masterclass. I have the pleasure to welcome Katarina Wimmer from the University of Innsbruck in Austria, who is a molecular geneticist and specializes in the diagnostics of hereditary tumor syndromes, amongst which NF1, of course. And who is going to give us today a very interesting lecture on the difference between NF1 and constitutional mismatch repair deficiency syndrome. So before we actually start the lecture, I just need to give you just a couple of instructions regarding the questions. You will not be able to interact directly with Katarina, but you can put all your questions in the Q&A box or in the chat box, and I will be moderating the Q&A right after the lecture. Thank you, everybody, and the floor is yours, Katarina.
So, thank you, Marco, for this kind introduction. I'm going to share my screen with you. And I hope you can see it now.
Yes, we can. Thank you, Katarina.
And I'm going to put it as a slideshow. So, yes, as you said, I'm very honored that I may give an masterclass on constitutional mismatch repair deficiency syndrome and its connection with NF1. And I will show that it can be sometimes a differential diagnosis to NF1 or Legius syndrome. So just to give you a short overview on my presentation, I will first give you an introduction of what is mismatch repair, what function it has. Then I will introduce the constitutional mismatch repair deficiency syndrome, short CMMRD, the genetics and the clinical phenotype, and then the overlap of CMMRD and NF1 phenotype. And we will then discuss when CMMRD should be considered a differential diagnosis to NF1 or as a possible differential diagnosis to NF1 in a cancer patient and in a suspected sporadic NF1 or Legius syndrome child in which we cannot confirm this diagnosis genetically. So what is the mismatch repair mechanism? So it's one of two mechanisms that safeguard that our DNA during cell revision is faithfully replicated. So many of you may know that during cell division, the DNA needs to be duplicated, and that is replication, and that is performed by the enzymes polymerases. And these polymerases make mistakes during replication, but they have a intrinsic proofreading activity, which is executed by the exonuclease activity of these polymerases, and they can correct the errors that they make, but still some errors escape this proofreading, and then we need a backup system, and this is the mismatch repair system. So the mismatch repair system recognizes the errors and corrects them then. I will not go into the detail how this is done. This is very technical. I just want to mention here that we have four important proteins in this mismatch repair system, which is MLH1, MSH2, MSH6, and PMS2. And these proteins are encoded by the genes which have the same names. So these mismatch repair genes, together with the proofreading of the polymerases, together make sure that after replication, the new DNA strand is nearly error-free. So less than 6 errors per diploid genome during a cell division. So the mismatch repair genes, the 4 mismatch repair genes, they work as, for instance, the NF1 or the spread1 gene as tumor suppressor. And individuals who have an inherited heterozygous loss of function germline mutation or pathogenic variant in one of these genes have a cancer predisposition syndrome, which is called Lynch syndrome. So if you have a mutation, most of the patients with Lynch syndrome have a heterozygous mutation in the more penetrant MLH1 or MSH2 gene. Less Lynch syndrome patients have MSH6 and PMS2 genes, but this is due to ascertainment bias because these genes are more penetrant, so they confer a higher risk to tumors. And the tumor spectrum of Lynch syndrome includes mainly colorectal and endometrium cancer, but also a number of other cancers. The lifetime risk I'm giving here, don't nail me down, depends much on which of the genes is affected by a heterozygous mutation and whether it's a man or a woman and which mutation is present. So this is just a rough estimation. So the tumors arising in patients with Lynch syndrome, they arise because one cell gets a second hit in the wild type allele, just as in the NF1 gene, and from this a neoplastic cell arises, which is mismatch repair deficient. And if this system doesn't work, of course, mutations can accumulate after replication. So the characteristics of mismatch repair deficient cells or tumors is, in most of the cases, that the proteins are no longer expressed and that can be detected by immunohistochemistry, and that these cells accumulate a lot of mutation that may be single nucleotide exchanges, mainly C2T, but also other single nucleotide exchanges, and also small insertions or deletions. And these type of mutations occur very frequently at so-called microsatellites. So these microsatellites are sequences of short sequences who contain repetitive elements. I show that in the next slide. So these repetitive elements can be a single nucleotide as shown here. So a strand of, say, 25 T's, for instance, but it can be also dinucleotides like GT, 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 or 3 or penta or up to 10 nucleotide repeat units. So during replication, what often happens at such repetitive elements is that there is replication slippage, and some of the repeated elements loop out. And then if the polymerases replicates the daughter strand, these two nucleotides are not replicated, and the daughter strand contains only 23 T's. Normally, this is then detected by the mismatch repair system and corrected. But if the mismatch repair system does not work any longer, and this daughter cell clonally expands, all the daughter cells will have only 23 T's at this locus, and we can detect this by PCR amplifying this locus and then doing fragment analysis to see how large the PCR fragment is. And then we do that not only for one microsatellite marker, but for a number of microsatellite markers, and compare normal tissue and tumor tissue of the individual. And if compared to the normal tissue, many of these markers show a shorter sequence, so a shift to the left here of these signals, then we know there is some microsatellite instability, so shortened repeat elements. And the rule is that we call a tumor microsatellite unstable if 40% of the microsatellites analyzed are unstable, show a shift, and stable if none of the microsatellites analyzed are shifted. So this microsatellite instability testing is a molecular test that indirectly tells us a tumor is mismatch repair deficient. So that's as much for the introduction of mismatch repair system. And I come now to the actual topic of this lecture, constitutional mismatch repair deficiency. And I show this slide because this slide shows the first publications which were published in 1999 of in two back-to-back papers in cancer research. And these papers described the phenotype of CMMRD for the first time. Both papers described the phenotype of children which were born to consanguineous, so related parents who were coming from Lynch syndrome families. So both of the parents were Lynch syndrome patients, had a heterozygous mutation in the MLH1 gene, and the child inherited from both parents the mutated allele. So both alleles were inactivated. So any cell of this child had a mismatch repair deficiency. And the phenotype of these children was that they very early in life developed malignancies, mainly lymphoma and leukemia. In this family, there was also a child with a medulloblastoma, a brain tumor, and a rhabdomyosarcoma. And interestingly, all the children who had this condition also showed signs reminiscent of NF1, particularly capiolae spots, but some of them also other signs like freckling and cutaneous neurofibroma. So today we are 25 years later, and we know much more about this condition, which is now officially called constitutional mismatch repair deficiency, has an OMIM number, and we know from about 200 cases that have been published that most of the patients, and that is in contrast to Lynch syndrome, have mutations in PMS2 and MSH6, so more than 50% in PMS2 and about 20% in MSH6, and only a few in MLH1 and MSH2. And the reason is PMS2 and MSH6 are more prevalent in the normal population, heterozygous mutations. So that reflects more the frequency of mutations in these four mismatch repair genes in the general population. We know now that the clinical phenotype of CMMRD is characterized by a broad tumor spectrum. The main tumors we see is hematological brain and intestinal tract tumors. I will come to this in a minute. But in essence, any tumor in a child could be potentially a CMMRD associated tumor. And nearly all patients have pigmentary alterations, and most frequently these are caveolé spots, often reminiscent of NF1. So you see here, for instance, a child with CMMRD who has a segmental distribution of typical NF1 caveolé spots and also freckling, but other patients and may have also other hyperpigmented features or skin patches or skin alterations. And some children have also hypopigmented spots. These bluish spots are very rare in CMMRD. It has been reported in many patient papers that usually the hyperpigmented spots are different than those seen in children with NF1 or Legius syndrome. But I think this may be recognizable in some patients by some experts, but often this is not. It is not distinguishable if a child has CMMRD or NF1 or Legius syndrome from the skin pigmentations I show you here on the right and on the left side, patients with typical caveolé spots in children with NF1 and CMMRD, and you may guess by yourself which of these caveolé spots come from children with NF1 or CMMRD, and also here some phases. I have no voting, unfortunately, but here is the answer. So these are all images taken from children with NF1, and these are all images taken from children with CMMRD. So how frequent are these NF1 features in children with CMMRD? So multiple caveolé spots, 2 or more, and other hyperpigmentations of the skin are very, very frequent, present in nearly all children with CMMRD. 6 or more typical NF1 caveolé spots are seen most likely in approximately half of the patients. And some patients really fulfill clinical criteria for NF1. So they may have 6 caveolé spots and freckling or other signs such as Lish nodules, which have been seen in CMMRD patients, cutaneous neurofibromas, plexiform neurofibromas, and there are also each one child with a tibial pseudarthrosis and a sphenoid wing dysplasia and optic pathway glioma, to my knowledge, has been found in more than one child already. So these children may have really a phenotype, a skin or other phenotype reminiscent of NF1, and that explains why in the past many of these children were misdiagnosed as NF1 children. So why do these children have these features that are reminiscent of NF1? I told you before, if you have a mismatch repair defect, and these children have in any cell of the body a defect in the mismatch repair gene, that leads to accumulation of all kinds of point mutations. And the NF1 gene is already in a normal cell, a highly mutable gene. And that explains why we have so many sporadic NF1 patients. Half of the patients with NF1 have no family history of NF1. So it's a highly mutable gene, and there is also some indirect evidence coming from cell lines that are mismatch repair deficient and from tumors that we see in these cell lines, often NF1 mutations. And we have one patient reported that was from the original families in whom retrospectively an NF1 mosaic mutation was found. So this mutation was not present in all cells, but only in a proportion of the cells. So it happened after postzygotically and probably due to the mismatch repair defect. So very recently, another paper was published of a patient who with CMMRD who had also clearly NF1 features, freckling here, café au lait spots, and the plexiform neurofibroma. And also in this patient, a mosaic NF1 mutation was found. Interestingly, it was a duplication of a C in this stretch of 7 Cs, which is sort of a microsatellite element in the NF1, in the coding region of NF1. This group, this French group, analyzed then another 22 patients and found in another 2 patients mosaic NF1 mutations. So at least about 10 to 15% of the patients may have indeed mosaic NF1 as a result from the constitutional mismatch repair deficiency, but others may have only localized in the café au lait spots NF1 mutation, possibly also spread 1 mutation, but possibly also other mutations that cause hyperpigmentation. CMMRD patients also have other non-malignant features that are not typically associated with NF1. The most prevalent are multiple developmental vascular abnormalities. You see that in brain MRIs, and it is reported that very many of these patients have that, up to 100%. Less frequent are other brain malformations such as agenesis of the corpus callosum or non-therapy induced cavernomas. What I think is probably underdiagnosed are pilomatricomas. These are benign tumors in the skin, which results from beta-catenin gene mutations, and it has been shown by the group of Eric Legius that really children with multiple pilomatricomas have also probably also secondary to the mismatch repair defect mutations in this gene in the pilomatricomas. Pediatric lupus, systemic lupus erythematosus is rare, and it's a little bit more frequent in CMMRD patients, and a number of CMMRD patients also have defects in immune globulin production, but usually this does not come to clinical attention. These are just laboratory findings. So I come now to the spectrum of malignancies that is associated with CMMRD. As I told you before, there are 4 main groups, mainly hematological malignancies. Amongst these, T-cell lymphoblastic lymphoma are the most prevalent, and they have a mean age of onset that is quite young, so 6 years around. And a little bit later in life, CMMRD patients often develop brain tumors. Amongst these, high-grade gliomas are the most prevalent, but there are also others, such as medulloblastoma, seen in CMMRD patients, and patients who reach teenage age or adult early adulthood will more or less inevitably develop colorectal polyps and colon cancers, and also later then also other Lynch syndrome associated tumors. And then there's a fourth group which lumps together all the other malignancies that we see in CMMRD patients. More frequent are sarcomas and embryonal tumors, but as I said, in essence, any malignancy may be a CMMRD associated one. So with the next slide, I would like to compare or show the overlap of the malignancies seen in NF1 and CMMRD. So optic pathway gliomas are typical NF1 tumors, which are rare in CMMRD, and so far we have never seen a juvenile myelomonocytic leukemia in a CMMRD patient, but it is also rare in NF1 patients. We also have not seen MPNSTs, so malignant peripheral nerve tumors in a CMMRD patient. So these are typical tumors for NF1 and really rare in CMMRD. However, rhabdomyosarcoma can be seen or can be associated with NF1, but can also be associated with CMMRD. We see it in both. And there has been reports of an association of NF1 with pediatric high-grade gliomas and also with secondary MDS and AML. But these are older papers, and at that time, CMMRD was not that well known, and these were all in patients who were not genetically confirmed to have NF1. So our current knowledge that these tumor types are more frequent in CMMRD really challenges the diagnosis of NF1 in these reported patients. So with this, I would like to come to my first take-home message. So we should be aware of CMMRD as a differential diagnosis in any child that is suspected to have NF1 or Legius syndrome and has a malignancy that is not typically associated with NF1, such as an MPNST or a JMML. And that should be suspected even if the patient has mosaic NF1, because we have recently learned that mosaic NF1 may not be so rare in CMMRD patients. In cases where we have NF1 signs in the child that has a rhabdomyosarcoma, we need to do differential diagnosis and find out whether the child has NF1 or CMMRD, because that is important for future treatment and surveillance. Our previous knowledge on the association of pediatric high-grade glioma and secondary MDS, AML, possibly also on neuroblastoma and Wills tumor is really challenged by our knowledge of these tumors being more frequently associated with CMMRD than with NF1. So of course, the NF1 features were going also in diagnostic criteria that the Care for CMMRD consortium, a European consortium, has developed for cancer patients. So these criteria should give our scoring criteria or a scoring system in which each malignancy that could occur in a child is scored with or is assigned 1 to 3 scoring points. And then additional features that point into the direction of CMMRD are assigned 1 or 2 scoring points, and any child who, any pediatric or young adult cancer patient who reaches a minimum of 3 scoring points should be suspected to have CMMRD and should be tested. So these are the so-called C4CMMRD diagnostic criteria. But features of NF1 or Legius syndrome may be present prior to the patient developing malignancy. And then the patient will often be tested for NF1, as it was the case in this first case who had been described by Manon Swerin. In this case, who had at the age of 3 years Schreckling and typical NF1 associated caveolé spots, NF1 testing, and that was really comprehensive testing, could not identify an underlying NF1 or spread1 mutation. So later on, the patient was seen again, and due to the consanguinity of the parents who were first cousins, it was decided that CMMRD could be a possible diagnosis in this child, and the child was tested, and indeed a homozygous PMS2 mutation was identified. This mutation was already at that time a likely pathogenic mutation, classified as likely pathogenic, so indicating that the child really has CMMRD, and it was helpful to ascertain this diagnosis with assays that test for mismatch repair deficiency in normal cells. I will come to these assays in a second, and they could confirm that this child has really CMMRD. It was then put under CMMRD surveillance, as it has been suggested also by the C4CMMRD consortium, but unfortunately between 2 brain MRIs, as I was told by Manon, the child developed glioblastoma and died, unfortunately, from this glioblastoma. So often genetic testing for the mismatch repair genes, in particular for PMS2, which is a difficult to analyze gene, may not give a clear result, because you might identify a variant of unknown significance or only a heterozygous mutation, and then such so-called ancillary tests, which have been developed in different laboratories in Europe and also overseas in Toronto, can be very helpful. These assays more or less all test for mismatch repair deficiency in non-neoplastic cells, so mainly microsatellite instability in non-neoplastic cells, and the assays developed in Europe have all been evaluated by in a large number coming of patients and controls coming from the CMMRD consortium, but also the logic assay, which is mainly used in Canada and overseas, coming from the International Replication Repair Deficiency Consortium, is also very much evaluated. So except for the GMSI assay, which is the most simple one, all are very sensitive and very specific and can confirm or refute CMMRD in a patient where genetic testing does not reveal a fully conclusive result. I just want to mention this assay, which was developed at the Newcastle University in collaboration with our lab by Richard Gell. This is the so-called constitutional MSI assay, CMSI assay, and it essentially tests also for microsatellite instability, but it analyzes about 32 mononucleotide repeat markers, so 32 microsatellites, and not with PCR and fragment analysis, but a certain method that relies on next generation sequencing, and the data are then evaluated and result into a score, a so-called CMSI score. And what you can appreciate from this figure is, note that this y-axis has not a linear scale, it's a non-linear scale, that the CMMRD patients have a much higher score than normal controls or Lynch patients. So this assay we use also a lot in collaboration with Richard Gallen at the Newcastle University in patients where genetic testing reveals an unconclusive result. And this assay is also very cheap and scalable, so we can use it also to screen for CMMRD in research projects. So CMMRD can be a differential diagnosis in a suspected sporadic NF1 child, Legius syndrome child, in whom genetic testing cannot confirm this diagnosis. But how often is CMMRD a differential diagnosis in such a child? It was calculated theoretically that it is very rarely, only in 0.4% of cases, differential diagnosis. And we did, together with Lidwin Messiaen from UAB and Richard Gallen, a retrospective analysis of a large number of patients, 735, and screened them anonymously for CMMRD, and found in this large number only 3 cases with CMMRD. So these empirical data more or less confirm what was estimated before. So given this very rare differential diagnosis of CMMRD to NF1, the question arises, should we test every child in whom the diagnosis NF1 or Legius syndrome cannot be confirmed genetically for CMMRD? Technically, that would be possible, but we asked this question in the C4CMMRD consortium and thought first we think about the potential benefits and harms. So, of course, a potential benefit would be that you can start surveillance before cancer development. We know now better than when this slide was designed that surveillance is helpful and works and leads to better outcome for these patients. The parents can be informed about their own risk for Lynch syndrome and the risk of a second child, which is 1 to 4, and make family plans accordingly. But I think one of the biggest potential harms is that if this is performed very frequently, that we identify many children in whom we identify a heterozygous mutation or a BUS, and that leaves us with uncertainty, and that is awful for all parties involved, for the patients who live with an anxiety, for the doctors who don't know how to manage these patients, and that should be really avoided. So therefore, the Care for CMMRD consortium thought of developing some recommendations which patients should be tested for CMMRD. And they said at least 3 prerequisites should be fulfilled. So first, the child should have at least 1 diagnostic feature of NF1, and if this is not 6 or more papillae spots, then at least 2 hyperpigmented skin patches should additionally be present. Of course, the parent should not have signs of NF1, and an NF1 or spread 1 mutation should be excluded really with comprehensive and sensitive testing. And then the child or the family should have one additional feature that points into the direction of CMMRD. So the parents are consanguineous, or there is already a diagnosis of Lynch syndrome, or there is a cancer of the Lynch syndrome spectrum in a close relative at an early age, or if there are two siblings who have NF1 features and the parents are not affected, and so on. In the patient, for instance, hypopigmented skin patches or atypical papillae spots, or pilomatricomas, multiple pilomatricomas would indicate CMMRD. So at least one of these features should be present, and then it can be discussed to test this child for CMMRD, and ideally this would be discussed with the parents, with a physician who is trained in clinical cancer genetics at the center, which has specific expertise for NF1 and related disorder, and there is a multidisciplinary team who has agreed to that. The lab who tests then for CMMRD should have appropriate methods to avoid potential pitfalls that mainly result from PMS2 testing. I told you before, I don't need to go into detail, but the PMS2 gene is a tricky gene to analyze, and you need appropriate methods to circumvent these pitfalls, either in-house or in collaboration with another lab. And it would be also important that ancillary tests that can resolve ambiguous testing results, genetic testing results, are available either in-house or through collaboration. So with this, I come to my second take-home question, or my second summary. So CMMRD can be a differential diagnosis in a suspected sporadic NF1 Legius syndrome child without a malignancy, in whom we cannot confirm NF1 or Legius syndrome genetically, but it's a very rare differential diagnosis. So I suggest to follow the recommendation of C for CMMRD and test for CMMRD only those childs who fulfill these criteria that I outlined. And of course, we don't know yet whether these criteria have really a good positive predictive value, a good negative predictive value, how sensitive and specific they are. So we need further studies to evaluate them. And if we identify children with CMMRD in the course of such an analysis, it would be important to follow these children very carefully and register them in a database, preferentially in our C4CMMRD database, so that we get more information on the clinical course of CMMRD in patients which are diagnosed before their first malignancy, which would be a more unbiased method. And we will also be able to evaluate better in these children the surveillance protocols that have been developed for these children. And with this, I would like to come to my last two slides. So first of all, I would like to thank all patients and their families who really were willing to collaborate and donate also sometimes some material for research. That is very important. I would like to thank also all physicians to collaborate with us, sending patients for the diagnosis and collect data and treat, most importantly, the patients with a lot of dedication. I would like to thank all members of C4CMMRD. So you see here a picture which was taken when we discussed the guidelines of swearing et al. And I would like to thank also the European Reference Network, Gentouris, with whom we develop now currently new guidelines for CMMRD, and my funding organization, FWF. So with the last slide, I would like to make some advertisement. We have been working to the C4CMMRD consortium together with the European Reference Network and Gentouris now for several years, more than 2 years, on new guidelines for the diagnosis, counseling, surveillance, clinical management, and quality of life. So really comprehensive guidelines that hopefully will come out soon, early next year. And so watch out for it at the Gentouris webpage, and there will be also a short version of these guidelines published most likely in the European Journal. And with this, I thank you for your attention. I'm happy to take any questions.
Thank you very, very much, Katerina, for this very, very thorough masterclass. We don't have many questions as yet coming up from the audience. I encourage everyone to come up with questions since we have Katerina here live to answer them for us. There is one question from Annette Backer, which I think needs to be contextualized a little bit. And Annette asks if she expects treatments of CMMRD to be different from the NF1 treatments.
Yes, yes. That's in. I should have said that during my lecture. Of course, that's the main reason why we need to distinguish patients with NF1 and CMMRD, because patients with CMMRD, of course, have first of all, other therapies would work at them. So, for instance, brain tumors of CMMRD patients don't profit from the usual therapy of brain tumors, but they need immune checkpoint therapy. That works better on them. And they have a very high risk to develop another tumor later on in life, so they need surveillance, and that is also important. And also the recurrence risk is different. So if you have a sporadic NF1 patient, the recurrence risk in another child of the family is different, or is almost not given. But if you have one child with CMMRD, the risk that the next child has CMMRD is 1 in 4.
Absolutely. And I was wondering, because there are so few patients, so few CMMRD patients, that I suppose there are very few tissues to actually study. But I was wondering if we knew, for example, that a CMMRD patient who ends up, because of this CMMRD, ends up being a mosaic NF1 patient, because I wondered if, the biology of the plexiform of that patient was different from the biology of the classic NF1 patients. If you had even further mutations in that plexiform that could actually lead to it being more difficult to be treated, or more prone to becoming malignant somehow.
Yeah, this is unknown currently. We discussed that, of course. That's the big question. Will they have a higher risk to malignicize? But we don't know yet. So far, no MPNST has been seen in CMMRD patients, at least to my knowledge.
Yes.
But of course, I suspect that the French group who is following this patient will carefully monitor this.
Thank you. We now have questions popping up. So there is a question from June Ortenberg, who thanks you for this fascinating presentation, and who mentioned the fact that you've discussed recommendations for classic NF1/Legius patients, but what would you recommend for segmental patients? But I think this was discussed.
If you have a patient with a, I'm not sure if I understand the question.
I think it refers to, I would ask June to reformulate the question if I haven't understood it correctly, but I think it makes reference to the fact that you may have a segmental patient is what I would call a mosaic patient.
Yes.
So if you have, so what would be your recommendations for somebody who appears to be a mosaic NF1?
Yeah.
In terms of further testing for CMMRD?
Yes, several patients with CMMRD have been described as having segmental NF1. Yes, that is true, but I would still follow the C4CMMRD recommendations, and not because it is much more likely that the patient is actually a mosaic NF1 patient just by chance and not to an underlying mismatch repair defect. So that is much more likely than the patient has CMMRD. So even if there is a segmental distribution of the caveolé spots and no mutation is found, I would still follow the recommendations and analyze CMMRD only if another feature of pointing in direction CMMRD is present.
Thank you. There's another question from Ignacio Blanco. Again, congratulations. And he asks, at what age do you consider a patient as a child to suspect a CMMRD? There's always this big debate, you know, when does a child stop being a child in terms of
yeah. Most patients with CMMRD who reach late teenage age will have a tumor. So I would say late teenage age, 17, 18, and the patient does not have a malignancy, I would stop considering CMMRD as a differential diagnosis. But of course, this is just my thoughts about it. I have no proof or any study to support this. We know some patients with CMMRD who have attenuated CMMRD, but often they have also less caveolé spots. So the clear, I would suspect, also no evidence supporting this, but my suspicion would be those patients who have attenuated CMMRD have not so much of an NF1 phenotype.
Thank you. Ignacio points out that they used to consider that after the age of 8, people should have at least two or more signs of NF for an NF diagnosis.
Yeah.
Another question from Jose Velasquez, who says, what would you say are the key points to suspect a case of CMMRD in an initial suspected cases of NF1 with a negative result in molecular testing? So which are, the most important features one should look at within all the ones that you have listed?
Well, I think consanguinity is an important point. At least in our population, we see that often that parents are consanguineous, and I'm always worried when I see consanguinity. Of course, a diagnosis of Lynch syndrome in a parent is a very strong sign. A diagnosis of another sibling, say about the same age, also having NF1 signs, is also very suspicious, because either you find the mutation, so either the parents are mosaic and the children have inherited the same mutation from the parents, but if you don't find in both children the variant, then there is a big suspicion of CMMRD. Yeah. Multiple pilomatricomas, usually you won't have a brain MRI, but people who have seen a lot of brain MRIs on CMMRD patients say these DVAs, which are frequently not reported by the radiologists, because DVAs are benign brain MRI findings, and usually the radiologist would not report them because they are irrelevant for the clinic of the patient. But if you have, I wouldn't do a brain MRI in such a child just to find the DVA. But if you have a brain MRI, I would always ask the radiologist if there are DVAs, and if there is one DVA, that is frequent. But if
Yes.
there are multiple in different areas of the brain, that is highly suspicious of CMMRD.
So you recommend to actually make sure that the radiologist is clearly asked to look for a DVA in case of a.
If you start thinking in such a child of MRD as a possible differential diagnosis, I would not do an MRI to find a DVA. But if there is for another reason an MRI, I would ask specifically the radiologist, can you look for DVAs?
Thank you. We have another question from Junira Chen, who asked if the cafe au lait spots in CMMRD patients, do they always have an NF1 mutation?
So I can tell only from one patient that we were allowed to take some tissue, and it was cultivated at UAB. The melanocytes were cultivated, and no NF1 mutation, no spread 1 mutation was found. But that doesn't rule out that there was an NF1 mutation, but this has not been tested very frequently to my knowledge. I know only of my own analysis together with Ludwig Messian, and there we could not find an NF1 mutation.
We have another question from Andrea Peterson, who asked that generally, as a geneticist, a pilomatricoma makes me think of myotonic dystrophy, which can also be preclinical. Should we use age as a differentiating factor for this?
So, but in myotonic dystrophy, you won't have cafe au lait spots, I assume. The multiple pilomatricomas are indicative of myotonic dystrophy, but this will be a different cohort of patients where you suspect that. So we are talking about patients who have NF1 features as well, or
yes, exactly,
who has a malignancy.
But if you were, I think the question was, if you were to use the pilomatricoma as one NF1 criterion, should you also use age as a discriminating factor at all? Maybe that was more the sense of the question. You were very clear that it's not, you can't differentiate just based on one factor. That's very clear.
Yeah.
But I think this attendee has wants to have a feeling whether, you know, if you were to look only at the pilomatricoma, then would you use age as a differentiating factor? No.
The answer to this
The answer is no. Okay, there's another question.
Are just something you see in children. You don't see that in adults usually.
Yes.
During life.
Yeah, because Andrea says he or she has a lot of patients that have unexplained cafe au lait.
Yeah.
And another question has come up from Eniko Pivnik. He says, in any documented case, who has a non-mosaic NF1 and independent from CMMRD, I would imagine that patient presents with usual and unusual features and malignancies. Would it be recommended to test for CMMRD even if the NF1 test is positive from blood lymphocytes? So would you recommend to test, you know, not to be, because I think that the sense of the question is the two could coexist. It could be both CMMRD and NF1.
Could be, yeah. Nothing is impossible in our world.
Yeah.
Yeah, it would be interesting to analyze such a patient. If this is a malignancy that is not very typical for NF1 and very typical for CMMR, I would test, yeah. If this would be a T-cell lymphoblastic leukemia, a lymphoma, I would test, yes. If this would be a high-grade glioma, I would recommend to do that, especially because when you do testing for NF1, if there's a high number of cells having the mutation, you may not be able to distinguish between a mosaic or a germline or a constitutional mutation. That might be difficult at some time.
Well, before we close, I actually have a question regarding this myself regarding these gliomas. Did I understand correctly that you suspect that many patients who were labeled as NF1 in the past and who actually had a malignant glioma were de facto not NF1, they were CMMRDs, and that I understood that correctly, that de facto in the NF1, in the bona fide NF1 population, there is not such a high risk of a malignant glioma.
That would be yeah, what you deduce from that. I think we need studies to do that, but I think that the old literature on NF1 patients where this diagnosis was not genetically confirmed could contain a number of CMMRD patients. So for instance, also for the secondary, where do I have my slide? So for instance, in this paper from Maris, where they reported secondary MDS, AML, some patients had typical CMMRD tumors as the first tumor. So I suspect a number of these patients had CMMRD, and also a number of the published pediatric high-grade gliomas. Just one thing, the first CMMRD patient I got from a person very experienced in NF1. And she said, I want, that was a long time ago in 2003, I think. And she said, I usually don't want to know what is the NF1 mutation because at that time it was very difficult to find it. It took a long time. But I want to know in this case because this patient has a high-grade glioma. It has a glioblastoma.
Mhm.
Then we didn't find a mutation.
Yeah.
But an experienced person in NF1 thought this was an NF1 patient.
Mhm. Yes. But this message is very, very important, I think. It's very, very important. Our time is up, and I can't see any other questions in the chat. Thank you very, very much, Katarina, for being with us today and for making very, very clear to all of us what the overlaps and the issues with these misdiagnoses are. I want to thank the audience as well for being very participative today. And this masterclass will be uploaded on the YouTube channel as soon as humanly feasible. And I look forward to seeing everyone at the next classes. Thank you again, everyone, and especially Katarina for being with us today.
Thank you for having me.
Goodbye.
Bye.